Well, welcome everyone to the membership meeting. Can everyone hear me okay? Thumbs up. Okay, great. Thank you. My name is Paul Hagan. I'm on the board of directors of the Capillary Parkinson Society. I've been on the board for about 5 or 6 years now. And it's a real honor to be able to do this today, especially with the program that we have in store for you with speakers from the Michael J. Fox Foundation. And it's our hope that it'll be very informative and helpful to you. I want to go over just real quickly a brief agenda of what will be happening this afternoon, and then a few announcements. Then we'll get directly into the video. We will be having, like I said, a few announcements. We'll go over the video from the Michael J. Fox Foundation. There will be a short question and answer period after the video, where you'll have an opportunity to ask any questions of Kimberly. You'll see Kimberly in one of your tiles, and she'll be talking in a few minutes as well. And then also we will have at the end a brief video from the Power for Parkinson's, a little exercise video for you guys as well. And that will be the end of it. It's our hope that you will really enjoy this, this time together. And let me get into a few announcements. I promise to keep this very short and sweet. Some of you are already aware of these items, but I thought I'd better touch on a few of them today. In the local Austin area, many of you are familiar with Freytag's Florist. They have entered into an agreement with us to, in celebration of National Parkinson's Awareness Month, to give a 5% discount to anyone that orders flowers for them during the month of April. So I will be giving out my my home address in case anybody would like to make a delivery later on in the meeting to me. That would be wonderful. If you would like to take advantage of that 5% discount, if you have a pencil down or whatever, there's a code you have to use, and it is FFCAPS. That's all uppercase letters. FF stands for Freytag's Florist. FFCAPS, those 6 letters, and you will get the discount. Also, under the Austin Community Blooms program, they are going to be giving an ongoing 5% donation to CAPS for every time you order flowers, even after April. So CAPS continues to benefit from this program, and we would appreciate your participation. This is much like, for those of you that have been around for a while with CAPS, this is much like our arrangement with Amazon Smile and Randall's. Purchases there also qualify for a discount, and we are benefiting from your purchases there as well. Upcoming events, April 20th, there will be an interview with Michael J. Fox. You have to register in advance for that. There's information about that on our website. And then May 17th through the 21st, the World Parkinson's Coalition virtual conference will be available through our website. There's information on how to tap into that. And as many of you know, one of our board members, Corinne Jones, who is an instructor at UT, will be speaking at that conference, at that the World Parkinson's Coalition conference. CAPS t-shirts are still for sale. Maybe next month when you guys log on, we'll all be wearing a CAPS t-shirt, right? But in any event, your t-shirts are still for sale, and they're available online as well. We encourage you to continue to join our membership section. This is something new, but that's all available through our website. There's instructions on how to do that. And through membership in our membership section, or joining our membership section, exclusive events and various local discounts are available as well. So something you might want to check out. Just click on the membership section, and all those details are available on our website. Everything I've mentioned is available on the website, capitalareaparkinsons.org. That's capitalareaparkinsons.org. You're probably all familiar with it. If you have any questions, please feel free to call Mary at our office manager. She's the one shaking her head right now, waving her hands, standing on her head. She's at 512-371-3373. And thank you for Mary for your contributions there and all that you do. Okay, let's get on to our program. I am going to briefly turn this program over to Kimberly. You see Kimberly Tronalone up in the waving her hand too. Kimberly is the advancement officer with the Michael J. Fox Foundation, and she will be kind of taking it from here. She will be telling you a little bit about herself, and also introducing the other speakers that will be on this video. So bear with me while I get this queued up, and we will start the video. Okay.
Yeah. Hi there, I'm Kimberly, and I'm joining you today from Brooklyn, New York. I started at the Fox Foundation in November 2019, so I've been working remote for most of my time with the foundation. As Paul mentioned, I'm an advancement officer on our advancement team, and during a typical year, I'd actually be traveling throughout what we call the Mountain Territory, which includes Texas, kind of right up the middle of the country to Montana. Of course, we've not been able to travel. We're really looking forward to getting back on the road. But a silver lining with all this virtual engagement is that in our time working remotely, we've been able to host some high-level virtual research updates featuring Fox Foundation leadership who don't usually have the opportunity to join our in-person support group presentations like we're giving today. So today, CAPS is actually going to stream a recording from one of these events. I'm so excited we got the green light to share it with you. We recently held this event for a select group of Fox Foundation community members. Our co-founder Debbie Brooks, and the Fox Foundation Chief Science Officer Brian Fisk will lead the conversation. And it will be moderated by our VP of Philanthropy, Veronique Enos-Kiefer. They go through some of the recent developments in the Parkinson's research pipeline, certain approaches and programs that they're particularly excited about, and what all of this means for the future of Parkinson's research. This is a really rich, in-depth conversation. Again, a bit of a silver lining. If we could bring Debbie and Brian with us on every presentation, we would, but there's only one of them. So I'm really glad we can share their insights with you today. And the video is not available on our website, so it really is a special opportunity for the CAPS community today. I do also want to preface, since this meeting will live on the CAPS YouTube page, that this recording took place originally live on March 5th, 2021. So if you're watching this later on, some of the stats and numbers or facts discussed may have changed. You can always check out our website for updated information, but just want to flag in case you're watching this later on. I'm going to be in the chat during the recording. I'm going to share my contact information. So if you ask a question in the chat or during our Q&A that we're not able to get to, please reach out. I'm happy to follow up. I'll also be trying to answer questions in the chat and sharing some links to relevant information that comes up throughout the recording, different programs or medications that might be mentioned. I'm going to go ahead and pop a link in. And before I pass this off to Paul for the recording, I'm also going to go ahead and I'll put a link for that Michael event taking place next week in the chat in case you guys would like to register for it. So with that said, I hope you enjoy the recording. We're so excited to share it with you, and I look forward to your questions.
If we haven't met, I'm Veronique Enos-Kiefer, Vice President of Philanthropy at the Michael J. Fox Foundation. It's my pleasure to welcome you today. I'd like to introduce two familiar faces from the foundation. Leading our conversation is Debbie Brooks. She is co-founder and executive vice chairman of the foundation. And joining her is Dr. Brian Fisk. Chief Science Officer at the Foundation. Hi, Debbie. Take it away.
Hi, everybody. Thank you, Veronique. Welcome. I hope you and your families are well. I'm going to start off with a little remembrance. Next Friday marks 1 year since COVID changed our lives everywhere, and of course at the Fox Foundation as well. It's the day that our team moved to a complete remote status, and I have to say, well, it's amazing to see how much our team has adapted. You know, one of the things that has changed is how we get to be together, not just with each other, but with all of you. And so while we'd much rather be doing this in person, I'm glad we at least have the option to pull together some information. These are times where it's important that we be able to provide updates and give you an opportunity to hear from us directly. And so today we're doing it all virtual, as you can see. You know, in the context of breaking news, we didn't want to wait, of course. So today we're here to discuss a couple of things. There were 2 recent trials that had news, and we want to put that news in context. Secondly, we're going to talk a little bit about what's behind the pipeline itself. And then we're going to give you a chance to hear more about how we're adapting and ask questions. And so with that, I'm going to start with my first question for Brian. Can we start by chatting about the 2 trials themselves? And tell us a little bit, you know, one at a time. For each, can you summarize what we know about the program's outcomes and then help us understand kind of where this leaves the field in regard to these 2 targets?
So there were 2 trials that recently reported, at least partially reported, I should say, some of their main outcomes in the last several weeks. One of these was a trial. It is a drug called cinpanemab, and it's developed by a company named Biogen. And they are developing a drug that targets, oh great, so we have the slide up here so you can kind of follow along. They're developing a drug that targets a protein called alpha-synuclein. And so for those of you who have maybe been with us for a while and heard us talk about the science and sort of progress in Parkinson's, you might hear alpha-synuclein quite a bit. It's a really important protein that's been linked to the disease. Pumps in the brains of people with Parkinson's, and we think plays a critical role in how brain cells might die in Parkinson's. So a lot of companies have been developing drugs against this. And so Biogen had one called cinpanemab. You can see it there down at the bottom of your screen in the slides. They were running a phase 2 trial. So they were testing the drug in people with Parkinson's and then a control group of people with Parkinson's who were not getting the drug. And they've been running the trial for a while. And we were obviously knew that they were going to be probably reporting some results soon. And a couple of weeks ago, they reported really in kind of an investor report. So this is how sometimes some of these companies report some of their information, unfortunately, is because they're required to for financial reasons. But it was revealed that they had decided to terminate that program and stop development of that particular drug. At this point, we don't know all the specifics why. They simply said in their reporting that the drug had not met its clinical sort of measured endpoints, as they say. And so didn't appear to be working in the trial that they had run. So we're going to try to find out more, of course, in the coming weeks and months from them. We have a good relationship with Biogen, fortunately. They're working on some other Parkinson's drugs, and so we know that they're still committed to Parkinson's, which is an important point and message we want to deliver today. And so we'll continue, of course, to work with them to understand a little bit more about that particular drug program and the decision that they made. So, well, maybe we stop here for a second, Debbie, because it might be worth talking a little bit about alpha-synuclein before we jump to the other trial, because I think there's a lot really to kind of talk about in this slide in particular, and about why this target is so important and why we continue to be so excited about it.
Go ahead.
All right. So yeah, so as you can see here, Biogen was really just one of a number of companies that. As you can sort of see on the slide, clumps of this protein alpha-synuclein we think are sort of pathologically critical and important for, if not causing the disease, certainly contributing to the disease, loss of brain cells that happens in Parkinson's disease. And so many companies for many years have been trying to develop these kinds of drugs that can go in and actually get rid of those clumps of the alpha-synuclein protein. Hopefully, and by doing so, slow the disease process down, slow the degeneration of those cells, and then ultimately, halt the disease process itself. So many of the companies are leveraging technologies. Actually, some of these approaches are similar to approaches being pursued in Alzheimer's disease, which also has a similar type of, and it's not alpha-synuclein, but a similar type of protein clumping that they think is linked to Alzheimer's disease. And so when it came to developing drugs for around alpha-synuclein, it was, an easy sort of transition to sort of pivot some of those platforms and some of those technologies to focus on Parkinson's as well. And so when you look at that sort of pipeline of drugs that people have been developing for alpha-synuclein in this case, you see a lot of similar themes, where a lot of those companies, Biogen, of course, was one. There's another company, Prothena Roche, I can talk about more in a second. A few others, AbbVie, Lundbeck, AstraZeneca. These are all groups that are developing kind of a similar type of what we can call an antibody or an immunotherapy approach to target alpha. So even though Biogen is one of those programs that ended its development, we know that there are a lot of these other groups that are also continuing. Actually, interestingly, Prothena Roche, who has a similar type of antibody technology, their drug is called prasinezumab. How they call these things and name these things is a science in and of itself. But their drug, they actually reported their results last fall. And even though they also didn't see quite the big, robust signal that they'd hoped to, they actually saw some interesting signals in some of the other measurements trial. And so they're actually continuing to develop their program and moving forward on that. So again, we continue to monitor and watch that program as well. And so even though Biogen has decided to end its program, we know others are continuing to move. So again, we hold out a lot of promise still around the alpha-synuclein despite Biogen's recent.
Okay, good. Yes, so this is a nice looking slide, and it's robust, and I think that's part of what we're going to want to help people see is that while we have disappointing results from one trial, that there are a lot of shots on goal, as Michael would say. So, okay, let's move to the GBA trial. That was also recently in the news. Can you tell us more about this target, GBA, and what's happening in the GBA field today, along with how you would characterize this trial and what we know about it so far?
So GBA is so there are a couple of forms of Parkinson's that are really driven by our understanding of the genetics of Parkinson's. And actually, alpha-synuclein is actually, believe it or not, one of those targets as well. It was actually mutations in rare families in the gene for alpha-synuclein were actually some of the first genetic links to Parkinson's discovered back in the late 90s. But since that time, we've discovered other genes that are linked to where mutations in those genes associated with Parkinson's. And GBA is actually one of the big ones. It's a gene, it stands for glucocerebrosidase, and it's a gene involved in essentially lipid sort of production and metabolism in the body. And it was found that people who have changes in their GBA gene that tend to disrupt that enzyme, so make it not work quite as well, seem to have a high risk for Parkinson's disease. And so because of that, and because that same gene is actually linked to some other diseases, Gaucher disease, again, and this is a common theme, you'll see the companies were able to very quickly pivot some of the technology and the platforms they were approaching for those other GBA-linked diseases to now move them into Parkinson's. And so we saw that happen also very quickly for Parkinson's disease, where a number of companies were able to move into this space. So recently, one of the big companies, Sanofi Genzyme, they had been developing a drug called venglustat. Again, you can see that here marked on the slide. This was sort of an interesting drug. It was targeting a slightly different mechanism to get at this, cellular pathway than some of the other approaches. And so it was sort of using a different type of sort of molecular trick for how it was trying to target this mechanism. They'd been working on this trial for a couple of years. It was interesting, and that was probably one of the first trials we saw enter the clinic for clinical testing that was targeting a specific genetic form of Parkinson's disease. And so that was I think kind of a pivotal moment, because until that time, most trials had really just sort of taken kind of your more generally common forms of Parkinson's disease. And this was a case where they were actually targeting at least a portion of their trial to these people who were carrying certain genetic versions of the GBA gene. And that was kind of one of the earliest sort of precision medicine trials for Parkinson's, as we like to call it. Anyway, so they recently also, kind of similar to Biogen, reported out in some financial reportings that their trial again appeared not to show any sort of positive change or signal with the drug, and so they decided to end that program as well. Again, just like Biogen, we don't have a lot of details yet on specifically what they saw. Just kind of all we know is the sentence or two they included in their press release. And so we're hopeful. Actually, I think they're going to be presenting at a scientific conference here in a few weeks' time. So we're hopeful to be able to get some more information about the specifics of that trial outcome at that point. But similar to synuclein, there are a lot of other groups that are still, I think, very invested in GBA as a relevant target for Parkinson's. And so you can see that here on the slide too, that there are a number of other clinical trials that are ongoing. Eli Lilly and Prevail, for example, have an approach to try to elevate the activity of the GBA protein in people with kind of a gene therapy approach. And so they recently, actually last year, came together. Prevail developed the technology and then partnered with Eli Lilly with a big investment last year to further develop this program. So again, we're excited to see that investment, of course, and also that partnership to move that program forward. And then there are a number of other partnerships and programs that are also moving forward on GBA. So again, similar to alpha-synuclein, we continue to see a lot of, I think, momentum here, despite the Sanofi recent news. And we will continue to see, I think, these trials move forward for GBA as well.
Yeah, and so even on this slide, you can see that we call out a couple of ALERT2 trials. There are other key genetic targets that are also being investigated in parallel. And again, these are all targets that are looking at disease modifications. These for disease modification. Do you want to say a little about some of these other key targets?
Sure, yeah. LRRK2, or LRRK2 as we like to call it. So this is probably one of the first genes really linked to the disease, kind of in the mid-2000s is when it was discovered, that I think really got people excited about the sort of these genetic forms of Parkinson's and what they could tell us about not only the genetic versions of the disease, but actually more broadly sort of the more common forms as well. And so LRRK2 was probably an area where, I'll say at the foundation, we got involved very quickly early on. It was around 2005 or so when the gene discovery came out, and very quickly we were funding work and trying to understand the implications of this gene. So when it finally moved into clinical testing, that was for us was really a milestone moment that we were able to sort of celebrate the movement of that particular genetic target forward. So right now there's two main players in this space. As you can see here is the company Denali, who's partnering also with Biogen. Biogen's, as you can see, is very invested in Parkinson's, which is great. So even though despite their alpha-synuclein trial results, we know that they continue to be very invested in the disease. And so they are partnering on a drug, an ALERT2 drug, a sort of more traditional small molecule kind of pill-like drug that is currently in a phase 1 trial. They've started to present some of the phase 1 trial results, and we're hopeful that they'll be launching later this year, we think, a next trial, a pivotal trial, as they say, which will really test whether that drug has any sort of clinical benefit in people with Parkinson's. So we're waiting eagerly for that. Biogen, again, also has a different approach, a slightly different, more genetic kind of gene therapy approach in a way to target the ALERT2 mechanism as well. So again, two companies that we really think are doing amazing work that are really moving forward, again, with this other particular genetic form of the disease, which, again, if we can show it works, and both companies actually, by the way, are looking not only at people who carry the ALERT2 mutation, but they're actually testing the drug in people who don't carry the mutation as well, because it's important to see if these drugs might actually have benefit. And we think biologically there's some rationale to suggest it would benefit in more common forms of Parkinson's as well.
A couple things come to mind on ALERT2. One, I think the gene was identified, by the way, by studying Parkinson's disease in families. So this is where participation was one of the drivers of innovation. But that was back in 2004, let's say, and it was in the clinic by 2017. So I just want to point out, that's a really fast move in our field. And 13 years to be in the clinic is pretty, that's the standard we'd like to see set, and it was supported because we had leadership funding to move the field on several different concepts around a new finding all at the same time. And to the extent that we can create blueprints for doing this work, I think it's an accelerator. We saw that in LERP too, but what really, what I think about is that's 13 years, and that included a moment where the field was sideways. And so I think it's actually a good example because, even today we see some disappointing news in one of the nuclear trials and one of the GPA trials. Actually find that this is the time where our work and what our team does is most important. When things are going well, people can kind of lose sight of all the work that's behind the engine to kind of keep it moving, all the different pieces that we're investing in to see something come together to actually get something into clinical trials. But stuff can go sideways. And, I think before things went to the clinic, there was a moment where that was an issue. At the time, Genentech was the active program that turned into Denali's work, and they had some evidence in their preclinical research that there was problems seeing bad changes in the lung in lung tissue in preclinical models, and this threatened the whole class before it was even getting to people. But the foundation stepped in, and maybe you could say a little bit about why, when things are going wrong, we could be playing such a critical role, or even if things are slowing down or there are common challenges, say a little bit about that LERC2 safety initiative, because I think that to me is this is where we shine the most when things are a little uncertain and there's a little bit of sideways activity.
Yeah. Great example. So this was, a few years ago before the LERC2 Denali program entered the clinic. And again, as you said, before Denali, the drug was actually being developed by Genentech, who then sold it to Denali. And we had been working with them for, a couple of years, mostly around sort of biology understanding of LERC2. And we knew they were, developing their drug, and they came to us, with some indications that some of the laboratory studies they had done, they were seeing some effects in the lungs of some of the animal models that they were using to test the drug that were, potentially problematic. We didn't really know what that meant at the time. We just knew that they were observing some changes in lung cells in the animals that they were testing the drug in. And so we very quickly, the first thing when you see anything like that is, okay, what does this mean? Is this, a potential danger? Either is it something related to the drug itself, or is this actually something that if you, sort of play around and muck up the mechanism of LERC2, you actually cause this kind of thing? And so we were actually able, and this was a real surprise even to us at the time, we've been working with industry for years, but, the idea of bringing multiple industry groups together and getting them to work together was actually kind of an unheard of thing. And so we were actually very, very quickly able to bring together, not only at that point it was Genentech, but we were also to bring in Merck and Pfizer and other groups to really sort of look at that finding and ask, okay, is this real or not? Because they all had sort of LERC2 programs or were interested in getting into LERC2. And for them to move forward, this was a question they had to get answered. Was this a non-starter now for LERC2 treatments? So we were able to actually create a consortium of that group. We kind of affectionately called it the LERC2 Safety Initiative to kind of give it some little bit more clout. And it really brought those groups together and allowed us to really think about, okay, what is this observation, and can we test it further as a group and bring in all that knowledge from the different companies about their own drug programs to really try to get more understanding around it. And after a couple of years of work and actually getting a number of studies done with not only the Genentech compound, but we actually were able to get compounds from the other companies to test together, not as a head-to-head comparison. No one was trying to compete with each other. It was all really to answer this basic, really important biological question. And so with that, we were able to actually put a lot more data around that problem, demonstrate that it really wasn't kind of the safety concern that we initially thought it might be. It's reversible. We knew that, it wasn't really something that progressed over time. It didn't seem to affect the actual breathing function of the animals. And so with that data in hand, the companies were able to move forward, and Denali's even mentioned how those findings really helped them make the case to move their treatment into the clinic. And so I think, like you said before, this was an opportunity where things felt like it was going sideways. We were able to work really quickly to bring that group together and really solve that challenge. And so we'll find out more, of course, with the recent trials, and if there's another sort of sideways moment, as you said, that's again where we shine, and we can come in and help understand, bring the groups together, understand what the issues are, and figure out what some potential solutions could be.
Yeah, one of the other things that comes to mind is this, the LRRK2 situation was an example of something sideways. I mean, when Genentech came to us, they actually said, by the way, we're out, and here's why. And so for us to be able to rescue that and then get all the groups together to benefit from the common interest in what could be toxic, and then, kind of give data to those companies so they could say to their bosses, we don't have to be out, I think was amazing. There are it's not the same, but there are some things that we are doing right now that are designed to help all the trials underway, even though they are doing things differently, i.e. they have different compounds going after different targets, but they have some common challenges. And do you mind just for a minute talking about how, so this isn't a sideways moment, but there's kind of there are two trains running in parallel tracks, and they're both, they need to kind of keep, we'd like one of the trains to get a little ahead, and that's our ability to visualize alpha-synuclein in a living brain. People hear us talk about biomarkers. This is a certain kind of biomarker where we can actually use neuroimaging to see and measure things. Can you just give one, I think is a good example of something that we're driving while all these companies, you've already seen a couple slides full of trials underway. What is something like our efforts around synuclein imaging? How is that helping, and why are we pressing so hard right now to be trying to crack that challenge?
Yeah, no, I think you're getting a really important point. So, I mean, there's you can have the most amazing drugs ever thought of in the world kind of in your hand, ready to start testing in people. And if you don't know how you can measure the impact of those drugs in people, then you're kind of, just shooting in the dark. And so, something we, caught on to very quickly and have been really invested in, really almost from the start, is this idea, again, biomarkers and other types of measurements. You can measure these different sort of pathological features of Parkinson's, and the ability to measure the actual clumping of the alpha-synuclein in the brain is a really, really critical one, and one we've been really working hard on for a number of years. It's not easy, trying to get something in the brain that can sort of bind onto those clumps of synuclein and then be able to visualize that with an imaging scan is not an easy thing to do. They've succeeded in doing it actually in Alzheimer's disease, which is amazing to see. The protein that they're looking at is slightly more abundant in the brain than alpha-synuclein, so that's kind of one issue we've been dealing with. It's just the amount what we're trying to detect is much smaller. But we know at least it's feasible, and I think that's given us a lot of hope. And so we've been investing in a lot. We've actually sort of, stole some of the experts in the Alzheimer's field to actually come work for us, in the Parkinson space, exactly, and sort of poaching them over. And sort of making sure that, making it exciting for them. Last year, we launched a large program, supported program to help support more of these groups that are doing some work. And so we know that there are, some people who've got some good leads. And so we are hopeful this year we might get some more data on whether some of these imaging approaches could work. But if we had that in place, I mean, we'd be looking at these trials now that are currently in development, and they'd be actually be able to measure whether or not their drug is altering the levels of alpha-synuclein in the brain, which we currently can't do. We can measure it in CS, spinal fluid and the blood and things like that, but that's kind of a proxy. We don't quite know what that means if you show a change in alpha-synuclein in the blood when it compared to the brain. So if we had that kind of measurement, it would be really, really huge, and that's why we continue to invest so heavily in that area as well.
So it's one example, and I think it's helpful to put that in the context of this really strong pipeline for disease modification. It's literally thrilling to see so many things having marched into the clinic for disease modification over the last handful of years, which is good news in and of its own. But we know that we are still lacking some key tools that would give all those trials a better shot, and that's one of them. We'll talk about some other ones later, I think. So, okay, let's shift gears a little. We've talked about alpha-synuclein and GBA just in the context of this recent trial news. We've talked a little bit about the rest of the portfolio around those targets. We've talked a little bit about alternatives like LRRK2. These are all things that are trying to, as Brian said, kind of engage in the underlying biology and see if we can do something that actually interferes with the disease course, something quite fundamental, and that's why it's called disease modifying. But I have to say, alongside that, we've invested in and we've really seen a lot of progress in the kind of treatments that help Parkinson's patients better manage their symptoms. Can you say a little bit more about that? I think this is an area where the things have not only gotten into the clinic, they've come out and been approved, and this has made a big difference.
Yeah, no, it's a good point. So, I know we'll have the slide here in a second. I think so. What's exciting to see here is that we're getting better year by year at helping people address the basic symptoms of Parkinson's. And so, when you talk about sort of your daily experience with a disease like Parkinson's, this is really where what we're trying to do is give you that sort of ability to keep your day functioning, keep you functioning during the day, allow you to do the things that you want to do and you need to do throughout your day. And that's, a really core part of trying to deliver new treatments for Parkinson's disease. Of course, while we do that, if we can also develop the actual sort of cures and the treatments that can slow the disease process down, of course, that's the ultimate goal that we have. But it's great to see here because, since 2014, 15 or so, we've had a large number of new approvals for Parkinson's disease, addressing a variety of different symptoms. In large part, kind of the traditional motor sort of movement parts of problems with that people have with Parkinson's disease, but more often than not, we're seeing some of the other symptoms being addressed too. So psychosis and drooling, actually, and something called orthostatic hypotension, a low blood pressure problem that some people with Parkinson's have that can lead to fainting and falls and things like that. So we're seeing drug makers really think about some of these unmet needs and develop treatments for them. So in the last year alone, we had 3 new approvals. You can see them there at the top. Ongentys is a treatment to help sort of improve your traditional dopamine medication, helping it to get to the brain a little bit better. Kynmobi is actually kind of a strip you put in your mouth that dissolves in your tongue to provide some dopamine medication that can help, especially when you have these kind of sudden off periods when your symptoms kind of suddenly, when your movement suddenly stops. So it's sort of a way of helping to rescue that. And Percept, which is another version of a deep brain stimulation technology that was approved last year. Interesting. 2021 has started off, despite the pandemic, has actually started off well. We actually had a prior drug, it's on here somewhere, Gocovri, which had been previously approved for dyskinesia, which is kind of a move, it's sort of the opposite in many ways of a slow movement in Parkinson's. It's when you take your medication and you get sort of that uncontrolled movement in Parkinson's, and Gocovri is a way to sort of address some of that. They actually got approval from the FDA just a few weeks ago to apply the medication also as a combination with your traditional dopamine medication to help reduce some of the off periods as well. So we're seeing, even in those cases, some of the existing drugs be improved on and sort of shown to be beneficial as well. So that progress continues. And sort of prior to approval in the clinical pipeline, we see a lot of other ideas too that are being developed. Some of them are tweaks in sort of how to get dopamine, better into people with Parkinson's, but some are also targeting other kinds of mechanisms that we think might be able to address some of the symptoms as well. So we see a lot of real interest, of course, in industry and drug makers, not only, of course, in kind of the more cure type treatments, but also in these really important drugs too that we think can help address the symptoms today.
Yeah, so we would need to be really sober about how hard it is to get new treatment of any kind, and we certainly can't dismiss out of hand the fact that 2 trials in important targets had disappointing news. But I think part of what leaves us feeling constructive and optimistic is that there's a really durable pipeline, both in disease modification, recent, and also additional things that are going after improved symptoms. And when you add that to recent approvals, I think it feels like Parkinson's has a lot going on, and that keeps us very, very motivated and excited. We interact, our team, not only, Brian and his colleagues, all the PhDs interact with researchers all over the world, and we have an industry partnerships team that does the same. And so we're interacting with pharmaceutical companies and device makers quite a bit. And this appetite to buy programs has just continued. And basically that means we're seeing an investment by the types of entities, mostly pharmaceutical companies, who can take things over the goal line. And that's big. We have this concept of a de-risking strategy. Like we're never, the overall cost of a trial. I mean, just in general, trials take something like 20 to 25 years in an indication like Parkinson's, and they cost over $2 billion. And by the way, that's just the ones that are addressing symptoms. No one's ever gotten over the goal line with the disease-modifying treatment, so we can't really say, oh, this is what it takes. It hasn't been done yet. But when you think about how many more players are in the market today, it's extraordinary. That's what you want, is that robustness in the field. And just in the last half of last year, there were announcements from companies buying programs that total over $2.5 billion. That is a stunning reversal from where we were certainly in the last 2 decades, but even 10 years ago. And so I think this is what leaves us feeling really encouraged about the state of the field, even though we would rather see a continuation of these programs going from, one phase to the next. Negative news or disappointing news is there, but there's a lot to be excited about, and it's going to keep us busy. I know that. Hey, Brian, what do you think is behind that optimism? Because it's my job. I guess I'm known for my optimism, and I think it's important. Our team is known for its pragmatism and its soberness. We're designed that way on purpose. But tell us more about what you think's behind that optimism and the investment, and say more.
Sure. Yeah, no, I think it's a good point. I think a lot, at least to me, a lot of the optimism and I think the interest in the companies, making deals and merging and trying to work together, I think is that our understanding of the science and biology of Parkinson's has really increased over the years. And a lot of this has been driven by some of the genetics. I know you hear us talk a lot about genetics, and so does that mean that Parkinson's is a completely genetics disease? No, it's not. But what genetics do provide is sort of an open window to look into the underlying biology of disease like Parkinson's and get an understanding of the kinds of things that can go wrong in the body that can only ultimately lead to Parkinson's, even in people who may not carry those specific genetic changes that are sort of the initial trigger for thinking about going after a particular biology. So, a good example is LRRK2 or LRRK2 we talked about before. When that genetic finding came out, we knew that there were people who carried that mutation who were at good high risk for Parkinson's disease. But as we've sort of peeled away that sort of biological sort of pathway, we're realizing that LRRK2 might play a role in cellular processes that might actually be relevant for other forms of Parkinson's as well. And so again, that's why I think as people see that biology and that increased understanding, again, driven in large part by some of the genetics, they're getting excited. Because if you're a company, what you want to be able to say is with some confidence is the thing I'm going after, the thing I've developed this drug to go against and manipulate or inhibit or accelerate or whatever you need it to do, that is really based fundamentally on sort of a clear biological role with the disease that I'm trying to treat. So without that sort of clarity, it's hard for companies then to kind of make that commitment, certainly that $2.5 billion commitment, and the billions more that it'll actually take to get some of these drugs to the market, to make that kind of investment. That's a high risk investment to make. So when they see that biology building and that sort of clarity of understanding of the mechanism build a little bit more, it can really excite them. Plus too, some of the other aspects of it's there's much more energy, and I think Ben played a good role in that too. Sort of understanding and connection into Parkinson's and how to get involved and how to get how to engage the Parkinson's community. It's much easier if you're especially a new company coming into this space to pick up the phone and call someone at the Michael J. Fox Foundation and say, hey, we have this drug, we're thinking about getting involved in Parkinson's. What do you think? And what are some first steps we should take? And so not having, I think, that resource would make a lot of companies harder for them to get involved. But now they know there's that sort of standing resource they can come to and get understanding of the science and sort of clarity on the science, be a part of some of the work we do, for example, around biomarker discovery. There's a lot of ways that they can kind of get really more involved and actually help sort of increase the confidence around some of their own drug programs. And for us, that's been a big part of I think, excitement and the incentive for companies to stay invested in PD.
You know, one of the things that's shifted a little as we've had these successes, and when I think about successes, one of the ways we measure it are the ways in which we would invest in an early stage idea or a mid stage idea and help take the highest risk where almost no capital was around and help an idea get more data around it. And then it gets, you know, the goal is that it gets enough data around it that it becomes attractive and that there's enough risk mitigated that a pharma company will step in and make a bet. And it always surprises people to think, you mean pharma, not every company is working in PD? In fact, 20 years ago, the number was basically 0, maybe 1, Amgen, and now we see this. And you know, pharmaceutical companies are kind of in the business of buying a phase 2 idea, you know, maybe a little earlier, maybe a little, you know, maybe, but that, you know, they want a good idea because they have the capital and the expertise needed to take things over the goal line. And when we see this de-risking, one of the things we appreciate is an idea no longer needs our money to get the data around it to compel the final investment action. But our money and our expertise gets activated in a completely different way. One of the things I've always found to be a great barometer is that a program gets bought, a company is now in Parkinson's. So now they're going to go to school. And as you mentioned, Brian, you know, we're one of their first stops. They're going to call and get information. And they want information about how to think about designing a trial. They want to understand where are the best sites in the country. How do we think about this disease? The nuance is not part of their existing organizational knowledge, and and then one of the first things they often do is they realize, wow, there's a whole ready-made infrastructure here for helping companies succeed in their trials. We talked a little bit about the synuclein imaging and why that tool is so important. And when someone's trying to design a trial, it's one of the things they wish they had. And so that's an area of interest. But we've also done other big, significant investments in looking for biomarkers, mostly in this case, markers of progression, things that are changing over time. Can you say a little bit about that? And then also maybe we'll get to the ways in which patients are so important in these processes, both to help in identifying biomarkers by participating in learning studies that do them, but also ultimately even participating in research. But go ahead and talk a little bit about more of these biomarkers. These are the kind of tools and needs that really are being developed to help increase the probability of success and mitigate risk.
Yeah, so yeah, biomarkers are really critical and key. And so, you know, there are a lot of ways you can think about a biomarker and how it's used. You know, sort of the classic idea is, you know, something you can just, you go into the clinic, you say, I have this problem, someone takes a blood test and kind of based on that can sort of diagnose you with whatever it is you have. You know, that sort of diagnostic type of biomarker. And, you know, for something like Parkinson's, we don't have a lot of that. Most of it is based on your clinical symptoms. We do have some ways to image the dopamine system in the brain now that are starting to be used as a way to kind of complement the clinical diagnosis. But we don't have sort of a blood test for Parkinson's yet, other than, you know, at least in some people, you can test for certain gene mutations, and that'll at least tell you potentially if they're at high risk for Parkinson's. But probably what one of the biomarkers or types of biomarkers we really desperately need are what one you mentioned is the idea of something that could track the progression of the disease. You know, so right now we can use kind of a clinician's scale, as they call it. It's called the Unified Parkinson's Disease Rating Scale, and it's sort of just a general way to measure the severity of your symptoms in Parkinson's. And those of you on the call who might have had, who have Parkinson's and had this done, it's a variety of tests and surveys you fill out and questions you answer, and it kind of gives you a score that gives you a sense of kind of where are you on your Parkinson's journey. And it's, a roughly useful tool for giving you that general sense of where you are, but it's not a great tool for looking at, the progression of the disease in any sort of consistent way. And so having ways that we could sort of measure that progression is really key. And so in I think it was around 2009 or 2010, we've been investing in just general biomarkers for a number of years since the early days of the foundation. But it was around that time, 2009, 2010, we decided we really needed to kind of ramp this up and do something a little bit more proactive that we were not seeing kind of happening anywhere else. And that was to really establish, a population of people with Parkinson's, newly diagnosed, and follow them over a period of time and actually see if we could measure whatever we can measure clinically, biologically, that might be changing over those, that first sort of 5 to 10 year period of the disease. And we called it the Parkinson's Progression Marker Initiative, which was a good title for what its goal was. And it really kind of, I think, set the tone for a lot of the work we do at the foundation around biomarkers, because it just gave us this rich resource now in its 10th year, following people, for all that time, understanding the different aspects of their Parkinson's. And so we have clinical data and imaging data and biological data, direct from patient data, and all this data is being made available in real time. Researchers can download it and analyze it. Millions and millions of downloads of the data have happened over the course of the 10 years. And so people are publishing on the data and really trying to understand, what are these changes that are happening in that period of people with Parkinson's disease. More recently, we've actually started to look even a little bit earlier. So, one idea is you tend to diagnose Parkinson's now when you come in with sort of the movement problems. But we've, been able to, especially a little bit with some of the genetics and some of the early clinical signs, sort of move that period up a little bit, where we can now identify people who we think might be at risk for Parkinson's disease based on, certain characteristics that they have. And that's allowed us to follow. We've started to follow a small group of them people over the last few years, but we're about to ramp that up actually into a larger effort to really see if we can identify people, this group of people who are at risk for Parkinson's. If we can find measurements in those individuals that help predict the onset of sort of full-blown Parkinson's disease, that would be huge, because then we'd have an opportunity to even maybe test therapies in the future in that group of people and see if we could actually delay the onset of their full-blown Parkinson's in sort of in the context of a kind of a prevention trial, if you will, where we could actually help those people never really experience the full-blown Parkinson's disease that we know today, which would be a huge, huge opportunity, of course.
People often ask, what can we be doing to move things faster? I mean, it's of course, the whole world has been on pins and needles waiting for better tools around COVID as a disease, in this pandemic. And then, how do we test it? How do we know what it is? What am I at risk for? What makes me at risk? Am I going to get what kind of version of COVID? How's it going to affect me? And then how can I get vaccinated from it? If you can't vaccinate, can you treat me? And these are all things that I think are elevating the healthcare dialogue. And a lot of those things do apply. I mean, it's not because Parkinson's is the same kind of illness, but the process and all the moving parts in the science that convert to medicine are present. And there are a lot of parallels, and I'm excited that people's dialogue and kind of conversation level around these things are improving. And the point I want to make here is this concept of you have this pipeline that's driving forward, and it's encouraging, and it's diverse, and it's got a lot of promise in bits and pieces. Right alongside it, we have so many things to be doing that could just in time kind of deliver better outcomes for one, if not all, trials. And so one of them is these tools for understanding what are we actually measuring, something that is challenging given how much heterogeneity there is in Parkinson's disease. That disease variability is it's a crusher when it comes to trying to figure out a path. And so these biomarkers, these objective markers, come in so many different forms and fashion, but it's a massive part of the work. And small gains in this area have been helpful already. Things that are coming out of the kind of classic PPMI study have already been integrated in the trials that are happening today, and that we're hoping for more of that. And then on top of this, these findings that Brian's sharing, this idea that in following people at the earliest stage of disease and then understanding how they're progressing and then learning more, independent of PPMI about who's at highest risk and starting to put them in a similar path and protocol, we are getting these hints and indications that we might be able to determine, kind of redefine when Parkinson's is starting. And that can help transform the field. So this is quite exciting. And as Brian said, this is an area that's getting relaunched, expanded, I really should say. So we will be I know that there's some people on this call who actually are in PPMI, the original PPMI. I think there might be a few people here who have already tried to raise their hand for the PPMI 2.0. That as this relaunches in the course of this year, there are going to be two components to that, and this is something you'll hear us speak to a lot, and it's an important part of this delivering tools and progress alongside these therapeutic programs. One is we're going to be looking to and educating people about what are these high risk profiles, who looks like they're at risk, and how can we screen people so that we can follow people who are at higher risk and understand if there's anything going on with these same biological measures and protocols that might indicate that they're getting PD, have PD, and also following the ones who don't. High risk, but not getting the disease, and understanding what's going on in that pre-Parkinson's kind of stage could be a real game changer. The second piece is we will now start looking once again for people who have just been diagnosed, just new minted diagnosed Parkinson's patients, and expand the number of people that we have this early disease progression information, this longitudinal look, because a larger data set is just going to let us draw more conclusions. So this is one of the resources that you see on the screen right now, is just this is an area where this kind of data just translates in real time to support all the trials that are already underway and the ones that are getting primed and inching closer to getting into the clinic. One above it there, Fox Trial Finder. This is just in general a place where anybody can go and set up a profile and see if there's a trial out there that you're interested in that's looking for you. And just a quick reminder, there are two kinds of trials generally: interventional studies, where you are brought into a study and you are going to be part of the process to test to understand whether something is safe and efficacious. And they're phase 1, phase 2, phase 3, and a lot of sub-phases around these. But these interventional studies are the way we go about understanding if new treatments can work. But there are a second group equally important, which are generally referred to as observational studies. And these observational studies are the ones that are contributing these real-time learnings that inform how we do the interventional studies. And so both of these paths are really critical, and Fox Trial Finder is a place where you can go to understand if there's some trials that might be a good fit for you. There's certainly a lot looking, and most people are shocked to hear this stat, but right now, if you're trying to do a trial in PD to either do an intervention or to increase our understanding about the disease, trial sponsors are lucky to recruit one patient per month per site. And when people ask what makes things so slow, I assure you, slow recruitment is a big part of it. So when people are wondering what can we do to speed things up, please consider checking out places where you might be a good fit to contribute to science. The third thing on the resource page here is an upcoming third Thursday webinar. This is a series, and this in March, our third Thursday topic will be around the Brain Boost conversation on thinking, memory, and Parkinson's. This is just a series. Each one of these is on a different topic, sometimes medicine, sometimes research. You can participate live, but we record them all. We have scores of topics on our website in archive. Thousands and thousands of people access this and find them helpful. And so if you're not familiar with Third Thursday webinars, there's a plug for the one that's upcoming, and I hope you go check out the information. Or if you're in conversations with us, we've probably said this to you and you think, I haven't been to see lately, but there's stuff in there that you're going to care about. Veronique, you're back. You want to help us with any questions?
Yeah, absolutely. Debbie and Brian, thank you so much for hosting this conversation. I so appreciate the level of detail. We have such a smart audience and a well-informed group, as you'll tell from the questions that they've asked. As you've been talking, questions have come through our moderator chat, our event chat, and there were pre-submitted questions. But I just I've just really appreciated all that you've shared already. I'm going to start, Brian, with a question for you. There's appreciation of that you've shared that there are multiple targets and many shots on goal, and this individual has asked, Are there any other key targets of note that you'd like to highlight? And particularly Debbie mentioned inflammation. That is something that they follow outside of Parkinson's also. Is there research happening for inflammation within Parkinson's disease that could be helpful for treating PD?
We obviously shared a couple of, handful of the leading targets right now, mostly because these were, targets where drugs are kind of in clinical testing now today. But there's just this massive, amount of information and other targets that are being worked on as well. And inflammation's an important one, and it's probably worth thinking about what we even mean when we say inflammation. And what we're really talking about is some aspect of the immune system and immune function that we think, in some cases, at least of Parkinson's, might contribute or play a role in the disease process. And so there's some evidence that suggests this is true, at least in some cases. There's a little bit of some of the genetic findings that are out there suggest at least some of the genes that are linked to Parkinson's might play a role in the immune system. Actually, there's some reasonably good evidence that LRRK2 might play a role in the immune system. We don't know if that's the reason why LRRK2 is linked to Parkinson's, but we at least know biologically that it might play some roles there. So all this data, what it's pointing to is that there's potential for going after the immune system as one potential target for Parkinson's. And so there's a number of therapies that are in development right now, a few that sort of moving into the clinic that are looking at targeting different aspects and different sort of parts of the immune system that are very complex cellular process, by the way. The immune system isn't just sort of one target in a cell. It's this whole system in our bodies that's evolved over millions and millions of years. So targeting the immune system isn't sort of a simple thing to do. But there's some good work that is starting to look at different components of the immune system that we think might be linked to Parkinson's, to see if we can mitigate some of that process, and in doing so potentially be a novel treatment for the disease. So yeah, so there's definitely a lot of interest in that area.
Thank you for that. So Brian, there are 3 questions that were related to genetics, and I think you could maybe look at them all together. The first one is, this individual's trying to wrap their heads around how genetic Parkinson's is. Is it the majority of families have another person who might get it, or is it really sporadic? Another individual says they do not have a genetic mutation, and they know that a lot of the therapies right now are coming out of what we're learning from genetics, and they wonder, will those therapies help them too? And then a third person has asked, when we do have a cure, or as new treatments roll out, are new treatments applicable to all, or are they more kind of tailored medicine?
No, a really important question. And I know we talk about the genetics as if we're implying that, again, Parkinson's is all genetics. And of course, that is not true, we don't think. I mean, there's a lot of ways you can look at this. So if you just ask people, do you have a family history of Parkinson's, which is often kind of one of the first things you tend to think of if you think about genetics of a disease. The numbers I've seen, 15% or so of people will tend to report a family history of Parkinson's. There's probably a lot of people who don't even know their family history enough to even know whether they have a history. But, at least in some of the studies, 15% is the number I've seen. Now, if you just look at the genes we know about, so it's like the LRRK2 and the GBAs and some of the other handful of genes we have, the percentage of Parkinson's we can kind of more specifically link to a known gene is less than 10%. So we haven't certainly even covered the 15% of family history yet. So meaning that there's probably still genes to kind of discover that are linked to the disease. So the real answer is, we know that a portion of Parkinson's seems to be fairly strongly linked to genetic forms, but the rest of the disease is sort of in this kind of unknown, or idiopathic is the fancy name for it, sort of version of Parkinson's disease. Now, when you talk though about the power of genetics, one way I like to think about it is, when you discover a gene mutation linked to disease like Parkinson's, what that does is that kind of gives you like a flag in the ground of biology to say that, okay, I know that this gene, and genes are basically just recipes to tell the cell how to make a certain kind of proteins. So I know this gene is saying that if you mess up this gene, and thereby the recipe for making this particular protein, and therefore mess up that protein and whatever function it has in the cell, that is one way you can get Parkinson's disease. So what you can do with that information is look at that change, that cellular change, whatever it might be. Maybe it's the gene mutation causes an enzyme. So this is actually the case for LRRK2, causes the enzyme, the protein, to be overactive. So we think the LRRK2 protein in people who have the mutation is overactive. It's acting kind of abnormally. You can look for that same change and the impact that change might have in other parts of the cell in everybody with Parkinson's disease, if you have the right measures. And we actually have gotten better at measuring that pathway more recently with some of the work that we've been funding. So you can then use that as a way to screen a lot of people and say, you know what? Okay, we know the people with the genetic version have this change, but do we see a subset? And in some cases, we are starting to see some signals of this, a subset of people who don't have the mutation, who also seem to have that cellular sort of disruption, that cellular change. And that kind of gives you, you know, a portion of the pie, if you will, of people who are associated with a LRRK2 form of Parkinson's disease, even if they don't necessarily have that mutation directly. And that becomes in the population of people that you can maybe target with some of the LRRK2 drugs. So we don't know if that pie is, you know, 20% or 50% or 80%. We don't know that yet. We're still working on the tools to really measure that, but you can start using that kind of concept. And so maybe there's a percentage of, you know, PD that is LRRK2 late related, and a percentage that may be more GBA related, and others. And we know all of them are at least related to alpha-synuclein, in the sense that the sort of clumping of alpha-synuclein seems to be a common hallmark of a lot of the disease. So we can kind of use that sort of, again, biological flags that we've planted in the ground based on some of the genetics to actually paint a broader picture of the biology and the cause of Parkinson's disease. And that allows us then to apply these drugs to a larger population. So I hope that answered the question.
Like genetics is teaching us what Parkinson's disease is, and that information is helping us treat the whole population. That's very exciting.
Actually, one thing I would add in here is that most of what we know about the genetics in Parkinson's has been discovered and appreciated over the last 20 years, and actually a lot more in the last 5 years. And so LRRK2 and GBA are the two most common, and as Brian points out, they're not wildly common, but they're common enough that we can actually find enough people to study it, which is very helpful. And we'll be looking for people who carry those gene mutations who don't have Parkinson's, because they're going to be very helpful to study. But one of the other things that we're aware of is if we can go to different populations around the world, because most of what we know has been learned from studying Parkinson's disease in Caucasians, mostly males, and there's more information because the genetic mutations can differ across different populations. And so one of the initiatives that we are working on this year as part of that is that we're actually going to be trying to screen, I think, 100,000 people who aren't Caucasian all over the world. And so we have partnerships with different, in different parts of the world to do this screening. And in classic common sense intention, but rarely executed, all of those samples will be brought to the US and all examined on one common platform, and all the data will then be released. It's standardized and released to the scientists all over the world. So this is a massive investment to really possibly, we don't know until we do it, but it's likely we will nominate all sorts of new genetic targets that may be brand new, something about this relative importance of different genetic targets by studying whole new populations around the world, which I think is exciting. And then secondly, you'll do this better justice, Brian, but we're talking about these genetic risk factors or genetic mutations as cause, but we also know that things that have nothing to do with genetics are at play because we see people where we can't identify any genetic contributions. So there's a big category of environmental risk factors, and that's a term because it can be anything you encounter in your life in the world, and you think of things you might ingest, but it's also a head trauma and things like that. And this year we're just launching or just funding a handful of new projects looking at environmental ways that we can study environmental data sets with Parkinson's in mind. And so I think just to often, we talk about one thing to kind of put context around it, and then we don't get a chance to let there are a lot of other things that are on similar paths, but that don't come up. And so important to say, I don't know if you would say anything more about that, Brian, but we are doing some work also trying to better understand data sets to look at environmental contributions to Parkinson's.
Yeah, and I think it's an important point. That sort of intersection of gene and environment, I think, is critical. And obviously in the early days, before 1997, when kind of the first gene mutations were linked to Parkinson's, we just assumed all of Parkinson's was kind of an environmental cause and something in the environment. And there was reasonable data to suggest that. We do know there's at least some data to support that certain kinds of environmental toxicants, certain pesticides we think are actually associated with higher risk for Parkinson's. So there's good solid data to support some of that. Now, to what degree environment versus genes versus some combination, that's I think a harder question to answer, and probably for every person it's a different answer to that equation. But I think we can't look at one without really considering the other. Even people who carry, for example, some of the gene mutations with Parkinson's, not all of them get Parkinson's disease. So there are people walking around with mutations in the LRK2 gene who are fine and live into their 80s and have no Parkinson's whatsoever. So something is modifying, their, risk for Parkinson's. It could be other genes, but it could also be environmental, lifestyle. It could be other things that they're doing, where they live, who knows. And so I think we have to look at this as kind of a whole equation and not just assume it's one or the other. So you're right.
Yeah, I mean, it's such an explosive and exciting time as we're continuing to learn so much about what Parkinson's is, that's putting us on this path for multiple targets, multiple approaches, having new treatments that get to the heart of what PD is and do more than treat symptoms. I mean, we're so grateful for symptomatic therapies, but getting us closer to it. So thank you both for that. Debbie, I'd like to ask you the next question. So Michael has a new memoir this year, No Time Like the Future, and it addresses optimism amid very real challenges. And I think this year, everyone on Earth has faced very real challenges, and in science, there have been challenges. So what's the Foundation's approach to challenge, and how are we leveraging optimism even now to keep science moving forward?
One of the things that in conversations throughout 2020 that our team thought deeply about was just how is the pandemic and these changes that have been, insinuated into all of our lives impacting our community. And our community, I think of in two blocks. They're obviously related. One is the scientific community. We are funding research around the world in all these academic institutions. We're shuttered and we had trials underway, just participants, kind of what do we do now? And of course, just the general population. We are quite, it was a constant discussion about for a population and a community like ours where isolation is already a possible challenge, and lack of mobility and not being able to be connected to your network and your care team and your family, that isolation could be such a challenge. And one of the things is we did really try to do some things to address that, beyond just pivoting as much of our programming as possible to virtual. We launched a new product that we're just it's off the ground now, called the Buddy Network, something we've had in mind for a while. And we just said, this is the time, even though it's just one connection, we are so often asked when we meet people face to face, I don't know anybody else who has Parkinson's. And there's often a like me question, part to that. And what we've come to appreciate that could be a lot of different things, that someone wishes they could meet someone else in a similar situation. It could be a patient who's interested in meeting another patient who lives in their community so they can have coffee once in a while. It could be someone who says, I want to meet someone who's in my field. I'm holding a secret here, and I'm a little worried that, if some people know that I have PD, they're concerned about professional considerations, and others just want to be connected to someone who's at their stage of progression, or thinking about deep brain stimulation. There are just so many different ways we've appreciated people want to know what like me means. We don't tell them, we hear it. But being able to make those connections and make those introductions, not just one at a time, but have a platform that can facilitate that. I think we're excited about that. And a lot of people raised their hand early and have signed up. We'll see how it gains traction, but that's a place to kind of meet someone quietly. And I'm excited about doing something like that. But in the course of the year, and what I think even something like Michael's book talks about, is I've been listen, we can't pretend that this isn't a hard time for everybody for the obvious reasons, and particularly for Parkinson's patients. Patients can be in trouble. This is hard. Having said that, I'm also so impressed at everyone's resilience, and so, and including the scientific community. So today, the things that I think we can count on for optimism is people have started right back up. I mean, the science feels like it's cooking. It's not 100%. Labs are partially reopened, but one of the things we worked hard at was to tee up money. I mean, last year, we knew that we were going to take a hit on our fundraising, and we kind of quickly adapted a complete dashboard process. So literally, as money came in, we could send it out, one grant at a time, and we were really focused on making sure all our grantees had the flexibility, but also the next commitment lined up so that they didn't have to lay off people in their labs. I mean, this is important. Momentum was going to be everything. But the kind of things that I see today is just a sense that people remain engaged. And there is this momentum. There is an opportunity to act. Some things are on hold, but there is, more people coming to the table. There are real on-ramps that make it possible for people to stay connected. And there's a lot of data. And I just think data drives how much and how fast our field can move. And so, one of the things that, in my conversations with Michael, and for those who've read his book, I think that optimism remains possible in the context of gratitude. And I can say that there's a lot that we can point to in the last 1 year and say, okay, that was hard, but I'm grateful that we're making it through this. And it doesn't mean it's been easy. And I think it's been particularly tough on mid and later stage patients and families. It's been a desperately challenging time, but I think that there's a lot to stay focused on. And I'm excited about that.
Thank you, Debbie. All right, Brian, there are multiple questions about biomarkers, but before I move on, I just want to say again, thank you. Thank you for Debbie and this community, for everything that you spoke about, for the resilience, for the optimism, for not disengaging. It's, as much as it has been such a challenging year, it's also been really moving to see what people do, what scientists do, what communities do, what we all do when we come together and stay determined. So I appreciate everything you added there. All right, Brian, there are multiple questions about biomarkers. I'm going to highlight two of them. Right in our chat, our event chat, an individual asked, how can you tell if drugs are hitting their targets in human trials, given that we don't have all of the biomarkers that we need? And how do you tell if a drug is working? So that's one of them. And then the other question is, so you share that the study of biomarkers is important to getting to new treatment and a cure. PPMI is expanding, which hints that it's working. What are we most likely to see first? A disease-modifying therapy or something to measure it? And how are those things working together? I know those are both different questions, but they're connected by the importance of biomarkers.
Yeah, no, that's good. So let's start with the first one. So how do you know if a treatment is doing what you think it's doing? And I'm going to just sort of differentiate that question from whether you think the treatment is altering Parkinson's disease, because I think that maybe touches a little bit more on the second question. So the first question is actually really critical. So when you are testing a new drug, you want to know that the drug is not only getting into the body, and there's some basic ways you can measure that, simply by just measuring blood levels of the drug, for example, something we call pharmacokinetics. You want to know where does it get in the body, how does it get broken down, things like that. So that's a clear, sort of obvious type of biomarker, and one we know how to do. Usually when a company makes a drug, one of the first things they develop is ways to measure the drug in the body. The second sort of type of measurement, though, you want is whether or not the drug is actually, as they say, engaging the target of interest. So if, in this case, let's use LRK2 again as a good example of this, you want to know that your LRK2 inhibitor, most of the drugs are trying to inhibit that protein, is getting into the body and inhibiting LRK2, the function of LRK2. And that target engagement, as they say, is a really critical measurement, because without that, you won't really know, one, if you're hitting your target, and two, to what degree you're hitting your target. So are you inhibiting it by 20% or 50% or 100%? Because that helps you understand then if you do see any clinical, sort of signals and sort of benefits, how much of the drug and how much inhibition in this case you would need to have. And so that kind of target engagement marker is really critical. And for a lot of drugs in the clinic today for Parkinson's, most companies have reasonably good ways to measure their drugs' actions, at least in the periphery. One of the challenges we have, of course, in Parkinson's is a lot of the action we're trying to measure is actually in the brain. We can do that sometimes by measuring things in the spinal fluid, which can help us understand whether the drug is getting into the brain, because the spinal fluid is kind of a fluid that bathes the brain. And so that's one way we can kind of get a sense of whether those drugs are getting into the brain and hitting those targets by measuring spinal fluid, which is why if you're ever in a clinical study and they ask for spinal fluid, please consider giving, because it's such an important fluid to be able to do that, and because it's a really important fluid that we need. So with that said, obviously once you know it can engage the target, is it actually changing the disease process? And that then gets back, I think, to the second question you had, which is, how long is it going to take to find that disease progression marker? And that I will have to say definitively, I don't know. It is a really hard question. I think if we had, it also depends on what, again, part of the disease you're trying to measure. So obviously we would desperately love to be able to measure alpha-synuclein in the brain right now. That's why we're putting so much effort and money into trying to find an imaging agent to measure alpha-synuclein in the brain. So that would be one big one. I think we're getting close. We have some good candidates that are, I think, in the process of hopefully moving into clinical studies so that we can actually start to see if these will work or not. We've had a few attempts in the last few years that didn't pan out, but I think with each one, we're getting a little bit better and a little bit more informed about how to develop this type of treatment or this type of measurement. But there are a lot of other things we wish we could measure too. So I think, you're going to see it's going to be a slow process. We are getting better at, again, measuring some of these sort of biological processes in people, including in some cases in the brain. So there are other types of imaging you can do to measure some of the other biology, and that we're looking at some of those as well, including inflammation. We have some investments to look at different ways to measure inflammation in the brain. So if you can put all these together, you might start to get, a toolkit of measurements that can help you assess some of these. Now, whether these will all be in time to help the current sort of crop of therapeutics that are in clinical testing today, that's a hard question. And if one of those treatments, obviously we don't want to wait until we have the perfect biomarker to start these trials. We should be trying them now still. So one of these trials could, show a clinical signal that looks good. And we, of course, would still want to keep moving on that while we continue to develop the measurements to make the next crop even easier to measure the impact of. And so I think you'll see probably the answer to the question is kind of a parallel processing where with each learning, we'll get a little bit better at it, we'll get better at it. And even if we see a drug that's working and we have to kind of, go hand in hand, then I think that will probably be the reality that we'll see.
Thank you, Brian. It's encouraging that there's so much attention around it, that PPMI is expanding, it's tripling in size. And isn't it a stat that the data has been downloaded by scientists 7 million times? So it's definitely being put to good use. All right, Brian, since we're talking, well, there's some more questions about other targets, other pathways that may be potential therapies. And there are 4 questions about stem cells, which I'll just sum up by asking, are there any promising developments in stem cell research? Is this a potential area for new therapies down the road?
Yeah, so we get this one a lot. And I'll sort of back up here and sort of do my stem cell kind of 101 quick primer. So when we talk about stem cells for a disease like Parkinson's, we're sort of thinking about it in 2 ways. And the way that most of us tend to think about stem cells is in the context of using stem cells, which are just kind of early immature cells that have the capability to divide and divide and divide and make more cells, and can be coaxed to then become certain types of cells that we want them to become. So in the case of Parkinson's, if we want to replace dopamine producing cells in the brain, we have the ability and sort of the laboratory recipes to make stem cells turn into dopamine cells kind of in a dish, and then you can use those cells ideally as a way to replace by implanting them into the brains of people with Parkinson's to kind of replace those dopamine cells that have been lost. So that idea of using stem cells to replace what is lost is a sort of a core, I think, concept that people tend to think about for stem cells for Parkinson's. Now, there's kind of another use of stem cells or general cell therapy, not always stem cells, that has to do with these cells we know produce a lot of kinds of factors, including some factors that impact the immune system or that provide kind of growth, sort of fertilizer, if you can think of it that way, for the brain, that we know just from biology can help sick cells not be so sick, might help them sort of live longer, things like that. And so there's a whole sort of body of the kind of stem cell world that is about using those cells to provide factors to help keep your existing cells from dying off, or at least, if they're sick, at least maybe help boost them up a bit to keep them healthier for longer. So there's kind of the two arms that I tend to think about when I think about stem cells. The second one's a little bit harder to talk about because it's kind of a bit of a fuzzier sort of landscape. But the first one, I think we're starting to see some progress. The idea of, can you make these cells into dopamine cells, transplant them into the brains of people with Parkinson's, and get some benefits? And for years, I mean, this idea is new. Back in the 80s and 90s, there were the groups that were using tissue to replace these cells in people with Parkinson's, and showing, at least in a handful of people, some potential benefits. Once stem cells were sort of discovered, they realized, using discarded tissue wasn't the way to do it. Let's use stem cells and kind of make them in the lab. And so now most of the field, of course, has moved to the idea of stem cells instead. So a lot of the time was spent working with the cells, learning how to make them into dopamine cells. That took a long process. We actually, one of the first funding programs we did as a foundation back in the early 2000s was to fund kind of a group of investigators to come up with the best recipe, sort of like it was like a bake-off in a way, I like to think of it, where it was sort of, okay, I'll try your different recipes, figure out which one produces the most kind of authentic looking dopamine cells, and that was kind of the goal. And actually through that, we were actually able to get a number of groups, including one of the leading groups in the world right now, to actually develop that recipe that's really become the standard and kind of been tweaked over the years. A little extra cinnamon here, a little extra spice there, but it's now become sort of the standing recipe for how to make dopamine neurons for Parkinson's disease cell transplantation. So we're seeing that field kind of emerge again. We're seeing them starting to move some of these ideas into the clinic. Blue Rock Therapeutics is one group that's actually publicly stated that they're hoping to start a trial this year with their cell replacement approach. So, and we think there are a few other groups out there that are moving in that direction too. So it's looking good, and that is an interesting area that I think we'll get some more information around. One last comment about it, though. It's important to remember stem cell replacement, at least in its current form, with the idea of replacing dopamine cells, is not a cure in the sort of classic context that you're stopping the disease and curing you of your Parkinson's. What it's really doing is just replacing dopamine cells that have been lost in Parkinson's, and giving you some of that dopamine cell production back. So it might help with your motor symptoms, ultimately. It might help with some of your symptoms, but it's not a cure per se. So I think it's important to think about when we talk about stem cells that we're not really talking about the cure for Parkinson's. We're really talking about yet another way to replace the lost dopamine in the brain. You probably will still have to take some medications, with your Parkinson's, and you probably still have some of the other quote unquote non-motor symptoms of Parkinson's. And so I think that's one thing to keep in mind when we think about stem cell replacement therapy for Parkinson's, that it may not be quite a cure-all for everything in its current form, but at least could be another option for people, if it's shown to be safe and effective, of course, another option for people to address some of the core symptoms of the disease.
Well, it's so nice to hear that stem cell research is alive and well and has been quietly making huge progress over the years, and that it sounds like it potentially could be used in combination with other therapies to sort of create a whole package. So that's wonderful. All right, this next question is for both of you, though, Debbie, you've spoken to this, but The question is, what have we learned from the pandemic? I think a lot of eyes have been on science. We work on Parkinson's research, but can you respond from a scientific perspective? Like, what have scientists learned about new ways to work in science this year in this new environment? And if there's anything else you would like to add about how the foundation facilitates progress in moments like this during a pandemic or when a trial doesn't meet its endpoints. Brian, what have scientists learned from this experience?
From the pandemic, or sorry, say it one more time.
From having to pivot quickly to work under these unusual circumstances. Like, what are scientists' working conditions like right now? How have they been able to keep moving progress forward throughout the year?
Yeah, it was, interesting. Obviously, in March, about a year ago, when everything, suddenly stopped, we very quickly, obviously, my team's, our daily job is to give money to people and help them through the research in Parkinson's. And suddenly, they were telling us they were not in their labs. They were at home, like all of us, sort of sheltering in place and trying to figure out how to navigate this new world. And over time, at least for some scientists, it was fine. It was a good opportunity to write those papers and analyze those data that they'd been sitting on. So there was at least a period of time where I think a honeymoon period in a way that people were able to sort of kind of, okay, well, let me finally get to that data set and actually analyze it. Let me download some PPMI data and analyze it, things like that. So we saw a lot of people kind of doing that initially. Luckily, eventually over time, at least some sites, some universities and some laboratory sites were able to establish kind of a way to get back to work, usually kind of reduced capacity, alternating their research team every other day. So we did see a little bit of it coming back, but I think, the researchers are as committed to the cause as any of us. That's their career. It's their job to go in and think about complex questions like diseases like Parkinson's, and they were just as eager to keep working as all of us. And from our perspective, we just were trying to do everything we could to make sure that they, were supported and could keep that going. We did things like extend all their grant, timelines by several months because we knew they would need extra time to finish the work that we'd funded them to work on. And so we wanted to make sure that they didn't feel like they had a rush to get a report to us, during the pandemic. So we were able to do a lot of things to help kind of keep them motivated and keep them going forward. And we also found that, a lot of our meetings, of course, that used to be in-person workshops, we had to do those virtually. And we found, because everybody was at home, they were super excited to stay engaged and be part of those conversations and just be engaged the whole time. So we didn't know what would happen initially, but I think everybody came together, and it was good to see that the research community was still moving forward.
I'll add a few things in here. One thing I think that is a step that we won't go back on is the role that telemedicine was able to play. So having a much greater acceptance and utility around so many indications, including Parkinson's, I think means that telemedicine is going to help be a better tool. That'll be really important for people with Parkinson's who live in communities who may never get to see a movement disorder specialist. This may now open up a little bit more access to specialized care, particularly for remote patients. I think that's important. Also, we were able to, I thought it was a nice proof of concept moment that we wanted to understand more about how COVID was affecting Parkinson's patients, and our Fox Insight platform, which has over, I think, 60,000 people. Not all the 60 are always active. So if you're in Fox Insight and you're not active, go back, re-loop. But we got a very fast response, and thousands of Parkinson's patients were able to answer questions to help us understand what was going on with them, both whether they'd been diagnosed, we got some information about that, but also what was happening in their daily life. And that was information that we can use as we think about how to be advocates in our policy business. One thing I think too, and Brian, I'll start this and you might be able to say more. Of course, one of the things that's very interesting about COVID is whether we're going to see any links to some of the neurological, lingering neurological aspects of COVID, and just this whole, even these handful of cases where that have been reported around the world where it seemed as though someone got Parkinson's right after they had COVID. that isn't necessarily how scientists see it, but I actually think we've already started work with the health, trying to stand up a way to be tracking the kind of lingering neurological challenges from COVID and understand them better. It's really interesting that one of the symptoms of COVID is this acute loss of smell, because we know in Parkinson's disease, there's often a gradual, imperceptible loss of smell over time. And, these are just things that probably, my suspicion is, we'll be linking ourselves to new learnings that come out of these these close relations. And there's nothing that we see today that says COVID causes Parkinson's, or Parkinson's patients are worse off or are more likely to get COVID, except for the fact that they may carry other risk factors like age and comorbidities. But this is going to be interesting. And I think we did see, Brian, isn't it right? We saw an increase in incidence of Parkinson's after the Spanish flu. Or neurological conditions. So I think that there's going to be not only more things to be learning, but there'll be more resources to go after this. There'll be a high energy around it, and that could indirectly or very directly bring new knowledge to our field, and vice versa. What we know in Parkinson's around some of these challenges. I heard a doctor on a morning show today talk about microglia, and I'm like, okay, the whole world has come around, So Brian, maybe say a little bit more about that, because I think scientifically, different than our operations, there might actually be some new scientific areas to be, some threads to be pulling.
Yeah, I think it's so. Last year there were some reports, and I think it's important to remember we've only really known about the coronavirus, or the SARS-CoV-2, the current coronavirus, for a little over a year, right? So we're still learning a lot about what it is and how it sort of looks in people and what it does to people. There have been at least some reports of neurological issues that people have had after having COVID that are being sort of followed up, and at least there were a few studies that were looked at that identified a handful of people. And when I say handful, we're talking about, I think there have been like 5 or 6 total descriptions of people who seem to have features of Parkinson's disease soon after having COVID. And so I think what that means in practice, we don't know for sure. And it's probably important to remember that Parkinsonism, which is kind of the more clinical name of the symptoms that are linked to Parkinson's, so the slow movement and things like that, and Parkinson's disease are kind of 2 distinct things. Parkinson's disease is a Parkinsonism. There are other things that have Parkinsonism-like features as well that aren't technically Parkinson's disease. So whether these cases are actually cases of Parkinson's disease in the classic sense, we don't know. Whether it could be that viral infection or some sort of brain infection induced by it led to loss of dopamine cells that give you the features of Parkinsonism. It's possible that could be true, but we don't know enough yet until I think we see more of these cases and understand what that means sort of broadly. And you're right, we are in conversations with a number of other groups that are, of course, very interested in seeing neurological outcomes post-COVID, who are working with the NIH and others to sort of establish at least some broad frameworks for how that information can be collected and followed over time, so that if we do start seeing certain types of neurological outcomes, at least we have some data to go into and start understanding what that means in practice. But right now it's still I think early days, and we're just continuing to stay monitoring it as we go forward.
There's a question from the event chat right here that 13 years is a lifetime for someone in their 60s or 70s who's living with PD, and that this is this is the timeline for getting to new treatments. And can you speak, both of you, either of you, to what is it that the field and the foundation are doing to accelerate the pace of getting treatments into the hands of patients?
I mean, let me take a stab at this. So I came to this field now 20 years ago with no appreciation for science or medicine. That's why I'm here, but I didn't have any. Trying to understand why things are so take so long and to kind of carry that frustration right into the room on my first days on the job. I have a lot of empathy for that question, even though I don't have Parkinson's disease. And, you can't get serious about changing that trajectory until you really understand it. And I think this was part of our DNA in the early days was to appreciate we're not just funding research. We are funding research, but it's in the context of what can we be doing to speed this up. And you do have to be pretty sober about what those challenges are. And so, it's funny, not too long ago, I was just re-looking at a video that I narrated probably 10 years ago now called How Do We Get to Cures? And talk about how long it takes for the aha moment to get to the drugstore shelf. It stands up. I mean, that's still legitimately the challenges we face. But the question's a fair one. What are we doing about it? And I'd say the main thing that we're able to do is, one, most of the players in the field don't see the whole field. They see a slice of the field. And one of the things we have done, and we weren't there on day one, but within a couple of years, we really had pulled back the lens, and we have worked very hard to appreciate the entire field and understand all the players. What are their motivations? What are their reward systems? What do they need? We've built relationships. I mean, when I say that we are at the nexus of activity, we purposely positioned ourselves there. And we remain there because we bring a lot of value to the field. But the main way we're executing that value is to kind of be sober, be really sober about why things don't work and go after them. And part of why things don't work is process. It's the things you think you can fix. You can see them and say, well, that's idiotic. Why don't we try it this way? And we have done a lot of those things. And in fact, although 13 years sounds long for that LRRK2 journey from discovery of a gene mutation to something being a target in being tested in humans, that's actually record time. And part of it was because we realized not everything has to be done in sequence. You could actually do some things in parallel. And just being in a position in the field to say, these things need to be done all at the same time, and we're going to launch them all. We're going to put money behind them. We know who the experts are, bring them together, convene them, and then provide the mechanisms for those groups to be speaking and learning in real time from each other. That's an accelerator. Being able to have set up granting programs where there's a lot of exchange of information. By the way, it doesn't help to tell Debbie Brooks. You need to tell Brian. Brian Fisk needs to be the one that hears about it. But then to not just say, that's interesting, but to say, that's interesting, what's next? Here's how we can help. Some people have tried this in other areas. Let us guide you over here. To be a constructive, informed partner who has a lot of resources to bring to the challenge of the moment. If you look at our portfolio of activities, there are probably 600 active grants at a time, actually more now, I think like 850 active grants at a time. But they're all different sizes, different durations, going after different things at different stages, which means our team has a lot of enterprise knowledge. And I think this is also an accelerator. But at the end of the day, Michael Fox is famous for saying biology's hard, and it is. And so we can tee up a lot of stuff. I spend a lot of time reminding patients and families, as frustrating as it is, there are actually some of the things that could be faster if patients were more engaged. And it's not just about funding the science. Funding matters. The more funding we have, the more things we can do concurrently. And that has definitely been the case in our trajectory. We've now funded well over $1 billion worth of research, and that early slide with all those drug approvals, our fingerprints are all over most of the ones that have happened since 2005. So that, funding matters and that scale matters, but there's some things that we can do all the work, tee everything up, and unless a patient shows up to get into a trial, or unless patients and family members of patients say, yeah, I'll go do a screening, I'll do some blood work, these are the kind of things that new knowledge can be a tremendous catalyst. So I actually think a lot has been done. We have a far more convivial and collaborative environment. Everything we do now, and this is a standard that scientists expect and tolerate, it's both, which is not only are we going to give you grants to do this work, and we're going to work with you as a peer to help be a problem solver and an accelerator. But all your data needs to be shared so everybody can move faster. And these are the kind of things that in concert, I believe, are capable of making things go faster. But there are just some things that we're going to try and we're going to fail at. And so more shots on goal is a strategy. It's a portfolio strategy. And that's a lot of what we do. It's ironic. Sometimes people assume that we're looking for the right answer. And I think it's really important to share that we have, over a dozen PhDs who are looking at all the possible answers. And we're trying to give each one a fair shot because, we don't want to place a single bet. We want to place as many sensible bets as we can and make sure they all have the best chance of success. And so that concept of acceleration, I know that 13 years is a lifetime. I also know and love people who've had Parkinson's disease for 30 years. So I know that it's more than a lifetime to, that there's time to really keep making a difference. And just in the last 5 years, we've had a lot of new treatments that I know are making a difference for patients today. So don't think that we're complacent by that time frame. We spend our day's work chipping away and trying new things and bringing others along in processes that we think can accelerate this progress.
Thank you, Debbie. This has been such a rewarding and worthwhile conversation. We have so many more questions. Questions about the microbiome. There's questions about exercise. We will reach out to all of you with an email. All of these questions are brilliant. Hence, we've gone on forever. There's a question about interpreting science news, that it feels challenging to know when one thing is successful or one thing has failed, how to put it in context. I feel like this conversation has gone far to sort of respond to that question, that we look at the big picture of everything that's happening. I do feel aware of the time. Bless you all for staying on so long, and for those of you who are listening to this part after. But I think we should go with one last question, and it's basically a personal one to each of you, Brian and then Debbie. Just what are you most excited about in the PD research and within the Parkinson's community? Brian?
Sure. I think, and the recent maybe handful of trials news notwithstanding, I'm still just insanely excited by what the therapeutic pipeline looks like for Parkinson's. So again, Debbie alluded to this earlier. When we kind of first started, there were a handful of sort of meaningful treatments in development for Parkinson's at that point. It did not end up panning out. So there felt like for a period of time there was this sort of emptiness in the pipeline that didn't seem to be filled. But in the last 10 years, we've seen all of these sort of ideas move into clinical testing in particular that are focused, again, what we feel like are on fundamental mechanisms underlying Parkinson's disease. So again, the synuclein trials, the ALERT2 trials, the DBA trials, a number of other trials out there that are currently active. And so, we're going to learn a lot. Not all of them are going to show the promise that we hope they show, but we'll learn from those too. And we'll continue to move those pipeline forward. But just again, the power of the science and the biology that's sort of pushing, kind of knocking on the door at the clinic right now is really, really motivating to see. And I just know, even looking, and one of the things my team does all the time is kind of continuously look at the broad landscape even before the clinical testing phase or the preclinical phase. And when you look at that pipeline, it's sort of bursting at the seams as well. So I think for me, that's kind of where the excitement lies, because I know all that science that we talk about is actually moving ideas closer to the clinic, and we'll just continue to monitor the progress of those.
For me, a couple of things that are specific to PD. We just recently had a board meeting, and in preparing information to kind of sum up where we were last year, what I see is now we meet almost 200,000 new people a year who come closer to being engaged in their Parkinson's journey, at least in by one measure, by coming and looking for information from the Fox Foundation. And what I see in that is an extraordinary amount of potential, because there's another handful of people around them who are getting more educated, they're appreciating that as complex as this is, there are lots of levers that we're working to pull. And this just gets me excited, and I think when you put that against a backdrop of a durable portfolio, it's a really important way to think about it. We may see more failures, but we now have statistically enough shots on goal that something should work. And that makes me excited. Then I like the fact that our world now is so much more science and medicine savvy, just having gone through this last year, and we will continue to do that. And the little things, they're not going to translate to a cure for Parkinson's tomorrow. But when I see this massive increase in people going to med school, and when I see, that they're like, we see interest in neuroscience, we're funding new movement disorder specialists. Those applications are growing like crazy. There's so many more ways where if someone wants to bring their energy and optimism and pragmatism to bear in this, there's a place for them. Our policy work is picking up. I mean, there's just a lot. And so there's a part of me that gets a little exhausted because I wish we could be doing more of it faster, but there is a lot going on. And I think that's encouraging to me. The thing that I'll be most encouraged about is that I personally hope to get a vaccine pretty soon. I can't wait to get back on the road. I want some hugs. I miss people. I hope today, we covered off on a lot of things, and I see the names of dear people on who are participating today, and just want to remind you there's more for us to talk about. So I hope today's a big conversation starter, in addition to just filling in this backdrop of new news and also just giving us a chance to give you a kickoff on where we are and what we're focused on this year. So, it's it feels really good to be able to share more and hear more from others, and see that people want to know what they can do and how to be engaged, and that we're here to kind of help with that. It's motivating for all of us. And so thank you for being here today and being part of it.
Yeah, this conversation has done me a world of good, and just spending this time with you. Thank you to all of you who are here for being so engaged in how to be part of getting to better treatments and cures faster. You are all absolutely part of this. And thank you for everything that you're doing in your communities, being a lifeline of connection, even under these circumstances. We know you are an inspiration to many. I want to just draw your attention, if you haven't found the COVID-19 hub on our website that talks about all the COVID news as it relates to Parkinson's specifically. And so that is there. And just stay connected. Again, we could go on for 2 more hours. We will spare you, but we do want to chat with you again, as Debbie said. And stay well, stay healthy, stay believing. I know that Parkinson's is better now than it was 20 years ago, that diagnosis means something different, and that there are new treatments coming through the doors, so many, and they're coming so fast, and that gives me a lot of hope. And I'm so encouraged also by treatments that are getting to, how do we stop this thing in its tracks? It's going to happen, and it'll be because of the partnership with all of us. So thank you, be well, and good night.
Okay.
So I know that was a lot of information, and I want to thank you all for hanging in and staying for the whole recording. I know we have a little bit of time now for questions, so I will happily take some. I also know it was a lot of information, so if anyone wants to let this soak in and email me later, feel free. Otherwise, please let me know if you have questions. You can put them in the chat, or I'm not sure how you typically go through them in your group.
Kimberly, I'll start it off. If just a couple of quick observations. You're not giving us a test on this, are you? Afterwards?
No,
okay.
No test.
Okay. This was just chock full of information. I'm sure each of us found bits and pieces of things, material to take away. I was particularly interested in the stem cell research discussion, which we've heard so much about it, but it seems like it's taken a little bit of a back burner lately. But now it's nice to know that that's still in the pipeline. Another thing was the proliferation of new drugs that have come along in recent years. I was not aware of some of those drugs, and it's nice to see what those drugs are and what they're targeted to address. And I think that's very important for our members to be encouraged and know what, if they're having specific problems, what drugs are available. And of course, their neurologists are working with them on that. But I did have one question I wanted to ask you, and you may not know the answer to this, but Brian touched on this at one point, and that was on the Progression Markers Initiative, where he was talking about whether people at high risk, studying people that are at high risk for developing Parkinson's. And I know a lot of our members probably are wondering, well, what about my family members? Or what about my immediate family, or my cousins, my children, my grandchildren, or whatever, or generations to come for that matter? Do you know how they're determining which people are going to be studied? In other words, those that are high risk, so to speak. Is it based on, are those people based on that they're going to be studying or following based on genetics, or genetic predisposition, or someone in their family, or is it environmental exposures, or both of those, or even more?
Yeah, I think that's a great question. And I know there are a few very specific kind of, I don't want to say markers, because we've talked a lot about biomarkers, and those are certainly a more significant flag, but there are certain things we've flagged within the larger Parkinson's community as ways you might be able to determine heightened risk. So we're working with those particular determinations for PPMI, and those do include people with a family history and genetic mutations, also those experiencing certain sleep disruptions and sleep disorder without a Parkinson's diagnosis, and additionally, of course, a loss of smell, which, as Debbie touched on, is a little tricky now because we're seeing that loss of smell also in those who have had COVID. So those are kind of the flags we look for when we're looking at people who are high risk, and the hope is we can, measure those certain, or take those certain biological samples that Brian mentioned, cerebral spinal fluid, blood, even skin, sweat, tears, really simple stuff from those people who are high risk, continue to check in on those things. And then if and when some of those individuals do receive a Parkinson's diagnosis, we can go back and look at all those biosamples we've kind of been analyzing through that whole journey and see, is something different with those people who ended up developing Parkinson's in that high-risk group compared to those in the high-risk group who never did develop Parkinson's, and where does that split occur? When does it occur? So the high-risk group is a really valuable group, and truly, a big hug and high five to all of them, because I feel that must be a bit of an intimidating thing to raise your hand for, saying, okay, I acknowledge I may be high risk for this, and then getting involved in research from there. So that's what we're looking at in a high-risk group, and that's why they're so important to this longitudinal study, because we can really hopefully see that evolution on a biological level.
Great, thank you. That's very helpful. I think it's helpful to a lot of our members to know that.
Yeah, glad to say so. And there's definitely a ton of info on PPMI, especially since we're expanding that recruitment this year on our website. I shared a link, but there's a lot more on there. If you jump on fox.org and just search PPMI, there'll be all sorts of exciting stuff. I do see a question in the chat from Stephen. The best way to follow up on progress on phase 1 trials. That's a great question. I am not familiar with any sort of systematic update that we get. I think in terms of the Fox Foundation, we certainly do quarterly check-ins with whoever we're funding and seeing how are those phase 1 trials going. are we moving over to phase 2? And we do put a lot of support into the phase 1 trials because, as Debbie mentioned, we're looking at that kind of high risk foot in the door where people aren't quite ready to throw big pharma money behind it. But if we can support it through that phase 1 point, make it really kind of exciting, we're seeing the progress, we're seeing this is valuable, then we get that pharma money and that corporate money to elevate it through to phase 2. So phase 1 is a focus for us. And I would comfortably say, I know we certainly track the progress of our trials and our pipeline reports and stuff within the organization. But I'd have to check in on, how do we really track that? How do we really look at that? I'm sure Trial Finder, the Fox Trial Finder tool, keeps all of that up to date since we are recruiting actively through that tool. But yeah, as far as a definitive, here's how we follow the progress, I think it varies for everyone depending on their funding, results, schedules. Someone could be really close to phase 2 and then, you go back and you analyze something and you got to retrace your steps a little bit or adjust from there. Not the most solid answer, but that's kind of where I'm at with that. Happy to dig deeper. Oh, as a layperson, how can you learn about progress? So we will certainly be posting updates. We love a good research update. In fact, our own staff website has suggestions of here are some great recent blog posts and research updates so we can share them out. So I would just check the website regularly. Our research team is always putting new information up.
Okay. Other questions, thoughts, comments? Kimberly, this is one of those presentations where people walk away from and they may be thinking, gosh, I wish I'd asked this question, or I wish I'd asked that question. Can you briefly just give us, again, one more time for those that weren't looking on the chat or whatever, your email address, so if that's okay, that they could get in contact with you?
Yeah, absolutely. I'll say it here. I'll put it in the chat again. And I'm going to share, like I said, the links that I put in the chat with you, Paul. And so you guys will have them on hand and feel free to share them out. And of course, feel free to share my email and info if anyone loses it or forgets. So my email is Tronolone, so Tronolone. You can see it on the screen. It's 3 O's. New York State thought it was 3 A's for a while. That was a fun fight to be able to vote. And my number is 212-509-0995, extension 273. So I'll pop that all in the chat. And yeah, if you all think of any questions, any feedback, anything at all that you would like to ask me later on at any point, please let me know. We're really here to support you guys and answer those questions. So even if it's a little more personal and less general, more specific to your Parkinson's journey, that's what we're here for. Okay. Yeah. Okay. Any other comments or thoughts? Thank you, Kimberly. Thank you so much for having me. Thank you so much for doing this. And I might mention this has been recorded and it'll be available for our members to see on our website at a later date. And for those that missed the meeting, so it was a great resource and a great update. Oh, I'm so glad to hear it. I'm glad you all enjoyed it. And again, I hope to see you in person next year. Great. Well, we'll take you up on that. Thanks so much. And please stick around, and if you'd like to, and we're going to go through a little video here exercise, and you're more than welcome to stick around as well. And again, thank you so much for all your time, and give our best to everyone at the foundation for the video and all of their work, their wonderful work. Our best to you and all your future work. Thank you so much. Have a lovely weekend. Okay, you do the same. Thank you. Bye. Okay, bye-bye. Okay, we're going to go through a little exercise video, and we hope that you will enjoy it, and bear with me as I try to get this rolling, okay?
steps. We hope you are doing well, and we hope you enjoy this short brain and body exercise sequence today. Make sure you have water nearby. It's important to drink water before, during, and after exercise. Maintain good posture. If anything hurts, don't do it, and feel free to make any modifications with the exercises. Remember that you can participate in Power for Parkinson's free live stream classes every day at our website, so we hope you join us. And finally, good news, we are hoping to resume our in-person classes by summer. Let's begin. Let's sit in our chairs. We want to sit forward in the chair, okay? So you're sitting straight, nice, good posture, and you're sitting at the edge of your chair. We'll take a deep breath, inhale, and let out a big ah, trying to hold it for 10 seconds. Here we go. Ready? Deep breath. Ah. Woo, and take another deep breath just to recover from that, right? All right, let's do it again, this time with a big E. Okay, you can smile while you do this one. Let's go. Deep breath. E. And that was very good. Okay, take a deep breath again and just be ready here with good posture. All right, we're going to rub our hands together, warm them up. Okay, we're keeping those abdominals engaged, okay, working the core. Kind of rub them together like you're washing your hands, something that we've been doing a lot of these last few months with that coronavirus, right? We're washing our hands nowadays more than ever. So scrub those hands. And let's do some angel claps here. Let's count to 20 by twos, okay? 2, 4, 6, 8, 10, 12, 14, 16, 18, and 20. All right, we're gonna make fists open, fist, open, fist, open. Get a nice stretch through your fingers, all right? And let's spell frog, okay? Here's frog, let's go. We have F, R, O, G. Now let's keep doing your hands close and open. Let's spell frog backward. Here we go. It's G, O, R, F. All right, open, close, open, close, open, close. Let's spell tree, T, R, E, E. Let's spell it backward. E, E, R, T. Nicely done. Okay, circle the shoulders up and forward, up, round, and forward, and go backward. And let's take that right arm scoop, go out to the side, front, and down. Left arm scoop, side, front, down. Right arm scoop, side, front, down. One more time, and scoop, side, front, and down. Very nice. Okay, so we're going to circle the wrists, and we're going the other way. And now bring one knee up towards your chest at a time. Just kind of gently pull it in. Kind of stretch for the hips and that low back. Alternate. We can take these at our own pace. And I know some of you need to increase the intensity of our exercises, and you go for it. That's awesome. Others might need to make some changes. Just make sure you're doing everything that's appropriate for you today, okay? And one more time. All right, let's lift that right knee, left arm, flicks here for the wrist and the ankles. 5, 4, 3, 2, 1. Switch. 5, 4, 3, 2, 1. Switch again. 5, 4, 3, 2, 1. switch, one more time, 5, 4, 3, 2, 1, and let's lift one leg out to the front, draw some circles, and then go the other way, and lower the leg, let's switch legs, lift, and then draw some circles, and go the other direction. And very nice. All right, put one leg out to the front. We're going to hinge forward. Wiggle your fingers a little bit. And come back. Let's switch legs. Hinge forward, neutral spine. Wiggle those fingers. And come back. Let's take a deep breath. Inhale. Exhale. One more time. Deep breath. Inhale. And exhale. All right, so we're going to continue warming up here. We've got a few little patterns of movements, okay? We're going to start by pressing front, back, front, back, front, back. Now, cha, cha, cha. So it's front, back, front, back, front, back, cha, 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 front, back, front, back, front, back. Cha, 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 front, back, front, back, front, back, cha, cha, cha. Excuse me, I have a little issue there because we are using music today, so I'm sorry for that interruption, okay? So now, the next exercise that we're going to do, we'll go out to the side. 1, 2, 3, cha, 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 and 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 1, 2, 3, cha, 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 one more, 1, 2, 3, cha, cha, cha. All right, the next exercise, down, down, up, up, shoulder tap. Down, down, up, shoulder tap. Again, down, down, up, shoulder tap. Down, down, up, shoulder tap. Down, down, up, shoulder tap. One more. Down, down, up, shoulder tap. All right, now we're going to touch the chest and open, okay? Chest and open, chest, open. We're looking up a little bit. Chest and open. One more. Chest and open. Now hold your arms here. We're going to march, march. Okay, now arms side, arms up, and wiggle your fingers, come down. So it's down, side, up, wiggle the fingers down and down, side, up, wiggle your fingers down. One more time, down, side, up, wiggle your fingers down. All right, we're going to climb the ladder, okay? Let's start one leg, right leg, left leg, and you're climbing the ladder, opposite arm as you're climbing the rung of the ladders. We'll count to 100 by 10s. Let's go. 10, 20, 30, 40, that's it, 50, 60, looking good, 70, 80, 90, 100. All right. Focusing just on the right leg, we're going to tap that heel out. So heel, toe, heel, toe, heel, toe. And pump those arms along with this, okay? Keep these going. Go on automatic pilot with these, okay? And I'm going to ask you to name at least 5 different animals, okay? One more. How'd you do? Did you get 5 animals? Let's do the left leg, same thing. Start with those heel taps. And we're pumping those arms. It's heel, toe, heel, toe. We're working the abdominals on this, arms, shoulders, legs, ankle, whole lots of muscles, okay? All right, while we do these, name as many musical instruments as you can. All right, let's go. And let's do one more. Okay, how'd you do on the musical instruments? All right, good. Okay, so we're gonna tap the right leg out to the side and the left leg. So one leg at a time. We're trying to make this as big as we can, so do your best. It's fine if we're going a little smaller. You do what feels right for you. And let's see if we can reach up with the arm at the same time. That's it. So we're going to count by twos to 21 this time, all right? And we're going to start with 1, and then here we go. So it's 1, 3, 5, 7, 9, 11, 13, 15, 17, 19, 21, and we are done with that, all right? So now go into your boxing kind of position. We're gonna punch across here, all right? Keep your abdominals strong, and as we punch, we have a little phrase to say, okay? So listen the first time and then join me. So here we go. My cat has fleas, my dog likes cheese. My cat has fleas, my dog likes cheese. My cat has fleas, my dog likes cheese. One more time. My cat has fleas, my dog likes cheese. Okay, we're gonna play the drums. Right arm, take it across. 1, 2, 3, slap. Left arm. 1, 2, 3, slap. 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 One more. 1, 2, 3, slap. All right, let's go through these exercises again, okay? And so start at the beginning, good posture, and we'll go front, back, front, back, front, back, cha, cha, cha. Front, back, front, back, front, back, cha, cha, cha. Front, back, front, back, front, back, cha, cha, cha. And front, back, front, back, front, back, cha, cha, cha. Now out to the side. Side, in, side, in, side, in, cha, cha, cha. Side, in, side, in, side, in, cha, cha, cha. Side, in, side, in, side, in, cha, cha, cha. And side, in, side, in, side, in, cha, cha, cha. Now palms, down, down, up, up, shoulder tap. Down, down, up, up, shoulder tap. Down, down, up, shoulder tap. Down, down, up, shoulder tap. Now touch the chest and open. Chest and open. Be as big as you can be. Chest and open. One more. Chest and open. Now march. March. Arms side. Arms up. Wiggle the fingers down, and it's down. And side, and up. Wiggle the fingers down, and down. Side, up. Wiggle the fingers down, one more, down. Side, up. Wiggle the fingers down. We're gonna climb the ladder. One leg, opposite arm. Get that knee up as high as you can. Counting to 100 by 10s. Let's go. We've got 10, 20, 30, 40, 50, 60, 70, 80, 90, 100. If you have not yet subscribed to our channel, please do. It would make us so happy. We're just so happy that you're here. You all have a beautiful day. Have a great rest of your weekend. Keep exercising. Exercise every day, okay? And we will see you next time. Bye.
Okay, well, that ends the meeting for today. Bruce and Jean, you get the gold star. It'll be delivered in the mail to your home addresses. For staying with us for the whole meeting, thank you so much to everyone that attended, and we hope you found it beneficial today. And we look forward to seeing you next week. Yeah, thank you. Thanks so much, and look forward to meeting you, Bruce and Jean, in a future in-person meeting that we hope to have.